Dear students, very good afternoon to all. Hope you all are fine. Today is our offline classes in continuation with our yesterday's offline class, that is derivation of DNA structure. James Watson and Francis Crick, on the basis of previous informations, proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was based on the observations of Erwin Chertoff. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence in other strand can be predicted. Thus, if one DNA strand has A, the other would have T, and if one has G, the other would have C. Therefore, if the base sequence of one strand is CATTAGGAC, the base sequence of other strand would be GTAACCTG. Also, if each strand from a DNA acts as template for the synthesis of a new strand, the two double-stranded DNA or daughter DNA produced would be identical to the parental DNA molecule. Silent features of the double helix structure of DNA are: number 1, DNA consists of two polynucleotide chains. The backbone is constituted by sugar phosphate and the bases project inside. Number 2, the two chains of DNA run in anti-parallel fashion with 5' 3' polarity in one and 3' 5' polarity in other chain. This is 5' 3' and 3' 5' chains. C is joined with G with 3 hydrogen bonds and A with T by 2 hydrogen bonds. Number 3 silent feature is the bases in two strands are paired through hydrogen bonds, forming base pairs BP. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. As a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of helix. This is the double stranded polynucleotide chain. You can see here one strand 5' 3' and another opposite 5 dash 3 dash glycosidic bond. You can see phosphodiester bond, and between C and G there is triple hydrogen bonds, and between A and T there are double hydrogen bonds. Also you can see the DNA double helix, strand 1 and strand 2. You can see shallow groove. The distance between pitch of helix is 3.4 angstroms. Then there is deep groove. Then you can see the phosphate, deoxyribose sugar. Helix length is 34 angstrom units. Here you can see again diagrammatic representation of the DNA molecule. Strand 1 5p-3' and 3-OH and 5-P. Strand 2. You'll see deoxyribose, phosphate, purines by double round circles. A for adenine, G for guanine. A single circle denotes pyrimidines. T for thymine and C for cytosine. And you can see the nucleotide. And you can see double lines and triple lines, they represent the hydrogen bonds. Number 4 silent feature is the double chain of DNA is helically twisted in a right-handed fashion. Number 5, each turn of double helix or the pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of a meter, that is 10 raised to power minus 9 meter. It has roughly 10 base pairs in each turn. The distance between two adjacent base pairs is approximately equal to 0.34 nanometer. Number 6 silent feature, the plane of one base pair stacks over the other in a double helix. This, in addition to edge bonds, confers stability to the helical structure. This was all about today's offline class. Thanks.